the lecture objectives are here. We're going to talk a little bit about formation through embryology, and then the different types of connective tissues going from through cartilage on into bone. And then when we talk about bone, there are different types of bone formation, intramembranous or endochondral formation, and I'll go over those processes. And then we'll do some pathology related to bone and cartilage formation. And there you go. So first of all, when you're looking at bone, it is a connective tissues, therefore it is derived from mesenchyme. We have the pluripotent mesenchymal cells, and pluripotent means that it can differentiate into any of the cell lines within that germ line. So that would include muscle cells, adipocytes, fibroblasts, chondrocytes, osteoblasts, any of those that you see up there. A pluripotent cell line in the ectoderm would give rise to either skin or nervous tissue, for example. And so endoderm would be linings of gut, vessels, and so on. So all of these things are related. All of these things are related. So during development, you start out mostly with a cartilaginous framework. And that cartilaginous framework gets replaced over time with bone. Now, for cartilage, we're going to talk about cartilage formation. The cell type that's predominant there is the chondroblast, or the more mature chondrocyte. These elements get replaced with osteoblasts or osteocytes to make the adult skeleton. To transform a cell from one of these mesenchymal cells into a bone-producing cell, or an osteoblast, requires certain genes to turn on. One of them in particular is the CBFA gene. When this gene does not turn on, it produces, or if you have a mutation in that gene, it produces all sorts of mutations or congenital anomalies in the skeleton. And one of them there is illustrated cleidocranial dysplasia. These people are born without clavicles and quite a few problems in their skull. Mentation is normal. They're just freaky looking people. And I ran into a person with this syndrome one time. He was not as a patient, but he was a hotel, the guy behind the hotel checking you in. Mostly every October, some buddies of mine, we go to Canada. And we stopped in some place in upper Minnesota for the night, and I go up there, and I'm tired, been driving all day long, and this guy checks me in. He looks a lot like that. It may have been that guy, but I mean, he reached for my credit card, and it's like, you know, his shoulders came together, and boy, I know what you have. You know, so that's the only person I've ever seen with the condition. But again, his cognitive abilities were spot on. So anyway, what happens is the gene starts turning on the osteocalcin, which is a protein synthesized by osteoblast that's required for mineralization of bone, and we'll talk about that later. When you see a defect in this gene here, and this mouse here with a blue cartilage matrix, if you have a defect, they can't form bone. And so this is the normal fetal mouse, and this is one with the defect, and they remain with their cartilaginous matrix. And it's never ossified. Okay? We'll talk about osteocalcin later on. First, though, let's talk about one of the most common cell types in the body, and that's the fibroblast We've talked about fibroblasts throughout your first year here. Fibroblasts secrete a lot of protein, so they're heavy protein synthesizing cells. Going to have a big nucleus, RAR, Golgi apparatus, because they're transporting it outside the cell. The protein product that they make mostly is a collagen. And so you see the collagen bundles here that are in the dermis. There's the epidermis right there. So this is all dermis. And you see how they're oriented in a certain plane. And what is that orientation called? When the collagen fibers are oriented in a certain plane?
Langer's lines.
Langer's lines. You should know that. So you don't make an incision in the wrong plane. You can see under a scanning electron micrograph how these collagen fibers look. They're quite numerous and they're quite thick. They're made out of protein. They're amino acids in a triple helix. So this is the base up there. You get 3 of them assembled here into a triple helix. And that triple helix, as an adult, is called tropocollagen. Tropocollagen is the functional molecule of collagen. It's not collagen. It's one molecule that will go to make up collagen. The thing you need to notice about this picture here is you see all these hydroxyl groups. Those are associated with 2 amino acids in particular, and that's proline and lysine. So a lot of the pathology we're going to talk about has to do with those hydroxyl groups on those specific amino acids, proline and lysine. The way you make this stuff, I think that's the next slide if I remember it right. Yep. Is the way you make collagen is you take a bunch of these tropocollagen molecules and you put them end to end. And these things, based on their amino acid sequence, they arrange differently. And if you stack them side by side, then what you wind up with is, this is a tropocollagen molecule here, and there's another one, there's another one. If you stack them side by side and you look at them under an electron microscope, they have different periodicities. Those periodicities have to do with where the gaps are between the molecules, the stacked molecules. The different periodicities describe different collagens. And the different collagens have different properties in terms of their strength or their pliability and so on. For example, the predominant collagen in your skin is type 2. The predominant collagen in bone is type 1. 90% of the collagen in your body is type 1. It's in bone. If you look at just the weight. You already know that type 4 collagen is found in 2 organs. What are those 2 organs?
Kidneys and
the lung and the kidneys. What's that syndrome? Goodpasture syndrome, right. So our antibody to type 4 collagen produces Goodpasture syndrome. You have, there are now over 30 different types of collagen in the body. So, all right, so what you do with these collagen fragments here, or these fibrils, if you want to call them that, a fibril. That would be a fibril right there. We're going to take a bunch of these fibrils and we're going to join them. And we join them with a protein complex. It's made of several different parts. One of them is, you see there's a string here. You see there's a string. That is a protein called hyaluronic acid. And then along the way of this height, now this is a protein. Those little dots are amino acids. Right? This is not, has nothing to do with carbohydrates. It has nothing to do with fats. These are proteins. So along the way, there are these linking proteins. These linking proteins right here. The linking protein you've got a core protein, and then off the core, you have chondroitin sulfate. So already you know that when you go to the health food store and you can even watch TV for joint strength and repair, what do they try to sell you? Chondroitin sulfate and hyaluronic acid. Why? Because they're involved in collagen, the stability of collagen. Anyway, these things link all of the fibrils together into the big collagen strand that I showed you in the earlier, in the first slide. Okay? Now, the way you make collagen is, since it's a protein, you translate the message into the rough endoplasmic reticulum. And when it enters the rough endoplasmic reticulum, excuse me, it's called pre-protein. Did you guys go to CAP today? They had this yogurt bar thing going on with the blackberries. And did you go over there?
No, I was at it.
Oh man, it was the bomb. I mean, anyway, and they were really sweet. They're really good. So pre-pro collagen is what gets translated into the RER, but as soon as it gets translated, the signal sequence on the end gets cleaved. And once it gets cleaved, it's called procollagen that arranges into the triple helix. Okay. From there, it makes its way on out to the extracellular space. Once it's, well, by the way, hydroxylation of proline and lysine occurs in the organelles as well. Hydroxylation of those hydroxyl groups that I showed you earlier, that occurs in the RER. You release this stuff, and the ends of procollagen get cleaved off. And once they get cleaved off, now you have a mature tropocollagen molecule. And from there, things get, they assemble and arrange, and there you go with the collagen fibers. Okay? It's that simple. Now, other components of connective tissue that fibroblasts secrete, one of them is elastic fibers. It's a proteinaceous fiber that has less strength than collagen, but more pliability. And you'll find it in certain tissues, like your ear here. Your ear here is, has more elastic fibers than does your skin. The skin of a 20-year-old has more elastic fibers than the skin of an 80-year-old. Reticular fibers are found primarily in lymphatic tissues, the tonsils, the spleen, the Peyer's patches inside the GI tract. Wherever you have lymphoid tissue, and they form a scaffold for the B and T lymphocytes. Okay? Fiber-related diseases, probably the classic one that's been known for a long time is scurvy, where you have to have vitamin C in order to hydroxylate the proline and lysine. So if you don't have collagen, if you can't hydroxylate, you don't have good collagen. Collagen is required for maintenance of certain structures. People with scurvy, where does it manifest initially? Where's scurvy? In your mouth, where you start losing your teeth because you don't maintain your gums. And in the skin, like the corners of the mouth here, chelosis, and there's no healing of, you have a wound, it won't heal. So not a lot of scurvy around these days in the U.S., but that's the deal there. Ehlers-Danlos syndrome has to do with the assembly of tropocollagen is jacked up because of congenital malformation of these enzymes involved in the pathway. Ehlers-Danlos syndrome is hyperelasticity and frequent joint dislocations. And the way these people usually die is usually by an aortic aneurysm somewhere. But the thing about Ehlers-Danlos syndrome is that one of the tests for it is to take you can take their thumb and pull it back and touch their, not that way, straight laterally, like that. That there's a lot of elasticity to their joints. Unfortunately, they usually, you have to screen these people and monitor them quite frequently because of the problem with ruptured aneurysms. Osteogenesis imperfecta is something that I talked about this morning quickly as a congenital malformation. It's a point mutation. You have an issue with the ability to form type 1 collagen. As I said, type 1 collagen is the primary matrix for bone. So these people, you see they're characterized by blue sclera. If anybody ever says to you blue sclera, the answer is osteogenesis imperfecta. That's the board question. But the problem with them is their bones are so brittle that they break. They break during the delivery. I mean, you look at this kid. He's like, you know, he's not. You can live into adulthood with this, but it's really a painful process. The calcium in bone is what makes it hard. The collagen in bone is what makes it strong. So if you don't have the collagen, the bones are like chalk. They're brittle. They're hard, but they're brittle. Okay. Goodpasture syndrome, we already talked about. Marfan syndrome, this is a defect in the fibrillin gene, which is associated with elastic fibers. These people have, one of the big things about them is aneurysms as well in their aorta and a lot of valvular problems in their heart. So they also happen to be typically tall, thin, gangly type people. They have a problem with dislocation of their limbs in their eye. The fibers that hold their limbs in place are defective, and they, I don't know, I can't remember the name of it, dislocated limbs, what that's called, but that's one of their problems. These people, they have, they're among us, and sometimes you're going to say, you know, that person looks like they have Marfan because they're tall, they just have a certain feature in their limbs are long out of proportion. They just don't look right. They're cognitively 100%, but they just don't look right. And Abraham Lincoln was thought to have Marfan syndrome, so it's pretty common. Anybody out here have a relative with Marfan syndrome? You got a couple?
Not a relative, but someone in my Boy Scout troop. He had Marfan's.
Uh-huh.
Yeah.
Was he characteristic, tall?
Yeah,
and
he's tall. He kind of leaned forward, long limbs. They just kind of like, you know, hung next to his body.
Yeah, it's odd. You see them, and so well, something's wrong with their limbs. They're longer than they seem to be. So anyway, any questions about that? Okay, formation of cartilage. Cartilage. So we've got fibroblasts over here, and then these cells are over here are going to differentiate into cartilage forming cells. There are different types of cartilage. We'll go over the three different types, three primary different types. But the way cartilage forms is up here on the top, you've got the just run-of-the-mill connective tissue up there, like the dermis, for example. Like the dermis, right here on your ear. So you transition from the dermis to the cartilage of your ear. That would be this transition you see here. These are fibroblasts up here. These are chondroblasts that are differentiating. These cells up here are secreting type 2 collagen because it's a skin. These cells down here are going to start secreting type 1 collagen because that's what cartilage is made of. As they secrete this stuff, they get surrounded by cartilage. Cartilage is not mineralized. Cartilage is pliable. It's not mineralized. Eventually, they get surrounded by this cartilage matrix, and they become chondrocytes. A chondroblast sits on the surface. A chondrocyte is surrounded by cartilage matrix. Cartilage is avascular. And these things aren't connected to each other, so you get nutrients to them, and they get rid of waste by diffusion. Therefore, cartilage can only grow to be a certain size. You can't grow a 4-inch piece of cartilage. Things don't diffuse. Cartilage has to be pretty small. If you think about in your body where cartilage is, there's, it's not really that big or thick. Okay? So a chondroblast is on the surface. A chondrocyte is surrounded by the matrix. And this is what they look like here. Once they're surrounded here, they're in a little place called a lacuna. So they're kind of cool. The surface up here where the chondrocytes are, chondroblasts are, is the perichondrium. That's the formative part to cartilage. The perichondrium replenishes the cartilage, the underlying cartilage. Okay. There are the three different types of cartilage that we want to talk about. The first type here is hyaline cartilage. If you'll see there, the number 1 is avascular. It says down here in the fibrocartilage is generally avascular. We're going to go with that. It's avascular. Okay. Type 1 collagen is the substance that each one makes. In elastic cartilage here, there is type 2. Type 2 is in the hyaline cartilage as well. Okay. Now, hyaline cartilage, this is what I want you to pay attention to, is where you find these different types of cartilage and their function. They're slightly different in their function. Elastic cartilage is obvious what it's going to be. It's elastic stuff. It's your external ear. It's your epiglottis. And although you can't see it, it's a eustachian tube. Hyaline cartilage up there is, it says right there, the temporary skeleton of the embryo. Remember that mouse where the cartilage was replaced with bone? The same thing happens to you. The cartilage is replaced with bone. That cartilage template is hyaline cartilage. It's also found in articular cartilage, not articular disc. I'm talking about the surface of a bone, not the menisci. That would be down here under fibrocartilage, or the disc in the TMJ. That's fibrocartilage. Most bones have a surface here on the end that's made up of hyaline cartilage. That's the articular surface. All right. And as important, the cartilages of the trachea, the rings of the trachea and the bronchi are hyaline cartilage. You get down here in the fibrocartilage, you're talking about the pubic symphysis and the disc, intervertebral disc, or fibrocartilage. Very, very tough substances. Okay? And the articular disc, as it says here, of the knee. Sternoclavicular joint right here between the clavicle and the sternum, that connection is joined by fibrocartilage. Not that it really matters that much. Okay. Now, the thing about cartilage is you see here in this bone, the hyaline cartilage forms the articular surface here. And the cartilage arranged, the cells that make up the cartilage, arranged in linear rows with fibers that are arranged according to the stress of the structure itself. And you see here how the fibers are oriented, they flatten out at the top, right? And if you think about that with bone, the same thing happens with bone, we'll see later on. If we look at this picture of the end of the bone here, this right here would be the hyaline cartilage. This is an area right here where the hyaline cartilage has degenerated. This would be degenerative disease of something. There's also a bone cyst there that's incidental. But you can see the difference. This is not good. That's healthy over here. So one other point I want to make on this slide right here is that when you're talking about hyaline cartilage and the articular surface, it does not have a perichondrium. It doesn't have a perichondrium. The perichondrium of this would be out here feeding the cartilage on the actual articular surface. The articular surface itself doesn't have a perichondrium. So once you rub it raw, it can't regrow. This is an x-ray of like a normal hip here. What you start seeing in degenerative joint disease is a breakdown of the cartilage and the bone. And it becomes, when I use the term feathered, that's what I'm talking about here. It's instead of nice crisp margins like you see the lip of the acetabulum there, it's just all smokied up. Okay? That's degenerative hip disease right there. That's going to have to be replaced. That will never get better. You can eat all the GNC nutritional products you want, all the chondroitin sulfate, all the hyaluronic acid. You ain't going to make that better. Any questions about cartilage? Pretty simple tissue. Let's talk about bone now. We'll start with intramembranous bone formation. Now, intramembranous bone formation is involved with, as it says here, increasing the diameter of bone. Not the length, the diameter. Endochondral ossification, that we'll talk about later, increases length. This increases the diameter. And where it says the increase the strength of bone is because there are two types of bone that are made in this way. One is called the cancellous bone or spongy bone, and the other is the compact bone. You can see these this right here would represent cancellous bone or spongy bone. If you carry this on down into the shaft of the bone, which is called the diaphysis, that would be the compact bone. Okay, we'll see it again later on. But you can see the spongy bone, how it looks in this illustration. Between all of these spicules, that's where you'll find marrow. That's where the marrow cavity is. Now, one way to form intramembranous bone is by a bone blastema. What you have here is you have a bunch of mesenchymal cells that, through a whole series of genes that turn on, transform into osteoblasts, essentially, and they become a syncytium. They join together and become a syncytium, and they start secreting osteoid. Osteoid is the substance secreted by osteoblasts. Osteoid. It contains type 1 collagen and a whole bunch of non-collagen proteins I'll talk to you about, I think, on the next slide. But what happens is they start laying down this stuff, and lo and behold, you wind up with a spicule of bone. On the outside will be osteoblasts. And just like cartilage, if they get surrounded by the osteoid and become mineralized, which will become mineralized, they wind up in a lacuna and they now become osteocytes. Just like cartilage, if it's surrounded by the matrix, it's a site. On the surface, it's a blast. So you can see these dudes right here, if they keep multiplying, they're just going to keep laying down bone. They're going to keep laying down bone, and they'll just lay down these spicules. They'll build these spicules all over the place, just as happy as they can be. Well, you got to stop that, or you'll wind up one big bone. So as these guys, osteoblasts, are laying down bone, there's another cell type called osteoclast, C-L-A-S-T, that will go behind them to eat bone. So let's go through that process here. Here's, you can see on the top panel there, a row of osteoblasts, and underneath it is a light-colored area up there, and then you can see it right there. That's osteoid. That's containing type 1 collagen and all of these non-collagenous proteins right here. One of those being osteocalcin that we talked about in the first slide. There's osteopontin, osteoprotogerin, and so on. Okay? Once the osteoblast lays down osteoid, calcium phosphate enters the picture and gets embedded essentially within the type 1 collagen. The calcium phosphate is the mineralization of bone. When I say bone is mineralized, that's the mineralization. This is bone right there. The black part up there is bone. It's mineralized, hard bone. Now, if you look at this cell here, see, I think most of what I just said is all you need to know. Osteoid, you don't need to worry about alkaline phosphatase, although I will say that an alk-phos, it's on a CMP. An alk-phos is on a CMP. Alk-phos is used primarily when you get to GI. We're going to talk about alk-phos being elevated in, if you have a stone, a blockage of the bile duct, it increases alk-phos because the bile duct tree, those cells that line the tree are loaded with alk-phos. So if you have an elevated alk-phos in somebody with gallstones, there you go. But alk-phos can also come from the bone. So if you have a bony tumor out here, you need to also, if you have an elevated alk-phos, you need to look at the liver enzymes. And you need to look at bilirubin, you need to look at albumin, you need to look at vitamin K depending clotting factors, like the PT, INR. Because if all of those are normal, you need to be thinking alk-phos is coming from the bone, not from the biliary tract. Okay. Sophia.
What did you say again? Liver function test, vitamin K,
I'm not sure I can repeat them all. So the ALT, AST, those are liver enzymes. They're not liver function tests, really they're liver enzymes. Albumin, vitamin K depending clotting factors that affect PT and INR, the International Normalized Ratio, INR. Prealbumin is another one. Okay, if all of those are normal, then you need to be thinking about, or maybe I better look at the bone here. Okay. Let's see, okay, so several non-collagenous proteins. One of them here that we're going to talk about here is called the ligand for receptor for activation of nuclear factor kappa B. That's RANK, rank, is produced by these cells. Okay? It's the ligand. Okay? Keep that in mind. We're going to go to the next slide. Yeah, we'll talk about parathyroid hormone in the next slide. Bone mineralization is a description. Do you have this on your thing? Okay, I can't remember when I updated it. This is a description of the mineralization process and how much bone is made up of collagen versus mineral. You don't need to know these numbers. That's just a description of the mineralization process, and I think you already know that. So let's go to this slide. Here's our osteoblast right here. There's the ligand, which is the L for rank. Now, in the marrow cavity here, you're going to have monocytes, tons of monocytes, right? Monocyte, you know, is an immature cell. Out in the periphery, they make macrophages. Well, they're used here as well. There is a receptor, well, back up. There is a product produced by the osteoblast right here. This binds to the receptor on the monocyte that causes the expression of rank. Rank binds to the ligand on the osteoblast. So now you've got an osteoblast sitting there on the surface of a bone spicule with a monocyte stuck to it. Of course, there are more things that go on with this relationship than probably we'll ever know. But what happens is these cells become multinucleated and large. They enlarge in their size. They eventually displace from the ligand, the rank ligand, and situate onto the spicule of the bone. Once they rest on the spicule of the bone, where they're touching it around their periphery, they start secreting proteins that seal the cell to the bone. That is called the sealing zone, right there. Then the part of the cell that's touching the bone becomes fimbriated. So you have all these little villi that you see there. With the discharge of essentially acid into the space that digests the bone. It demineralizes, it dissolves the bone. I'll talk about that in just a second. Let me back up. So this right here is an osteoclast. Osteoclasts come from monocytes. Osteoclast must have an osteoblast in order to be made. Well, what does Okay, so osteoclast is going to digest bone and release calcium and phosphate back into the circulation. So calcium is going to go up. Serum calcium goes up. What is the hormone that causes serum calcium to go up? Parathyroid hormone. Okay, let's stop it. Okay, here we go. Now go to the other side of this slide. There is a protein produced by the osteoblast. It's called osteoprotogerin. It has a greater affinity for the rank ligand than does the damn monocyte. And it essentially blocks the rank ligand and prevents this union. If it prevents that union, you can't increase calcium. So one way to decrease serum calcium is to produce that protein. Follow me? Well, wait a minute. Here's parathyroid hormone right up there. What does it do? It increases the expression of rank. And it also shuts down the expression or the synthesis of osteoprotogerin. It increases the number of rank ligands that will increase this relationship, increase the number of osteoclasts, increasing your calcium, serum calcium. And that's how it works. Pretty cool, huh? You know, it's a lot to take in, but if you just follow this 1, 2, 3, and then go to 5, you'll be fine. And then you can come back over here and do 4 and 5 over here, but go 1, 2, 3, 5. Oh yeah, that makes sense. And then you got 6, and then you can come back to 3 and 5, 4 and 5. All right? Very simple. I love these charts. So here's what this little booger does. This thing, as it digests bone, produces a defect in the bone. That little defect in the spicule is called Howship's lacuna. Howship's lacuna. I don't know who Howship was, but I'd love to meet the person. Because that's a cool name. Howship. My name is Howship. So anyway, the way this works is, you remember the equation. It's just like blood gases. You've got water and CO2. Now you got bicarbonic acid, and then you've got hydrogen and bicarb. That's the same thing happens here. There's a pump that dumps out hydrogen ion. You got a chloride exchanger. You got hydrochloric acid. In addition to hydrochloric acid, you've got enzymes that will digest the non-mineralized components, or the collagen essentially, that's in that it's being exposed to. The reason you've got this guy with this sealing zone here is to keep this crap from getting out into the bone. I mean, look at the pH of this stuff right here. Your bone, you know, that's not good to have out of your marrow. So that sealing zone keeps all of the bad stuff in Howship's lacuna. Very active process that goes on right here. Okay. So if we look at now developing bone, here's a spicule that we saw in that earlier, the bone blastema. Here are the osteoblasts, there's the osteocytes there. Bone is unlike cartilage in that you can't diffuse stuff through bone like you can cartilage. In order to keep these osteocytes alive inside of the bone, they have to be connected to the outside or to the surface. And if you look at bone, all these little, so there would be an osteocyte right there, there'd be another one there, there's another one there, another one there, another one. They're connected by all these little canaliculi that are in the bone. The processes from one osteocyte fuse with the processes of another. So they transfer nutrients back and forth through themselves. They're also, when you get down here into the lacuna is full of liquid that gets there through these canaliculi. That's impressive, huh? You guys are not impressed. I'm impressed. You'll be impressed one of these days. Okay? And here's the process that we've seen earlier. Osteocytes, osteoid, the bone, osteoblast. You can see up here, osteoclast. When osteoclasts work, they remodel bone. And bone is constantly being remodeled. It remodels based a lot on stress. If you start walking, you put more stress on your femur, so you're going to grow more spicules to handle the increased load. If you do weight training or something like that, you remodel your bone for your condition. That's one of the problems with space travel, is there's no weight that requires remodeling. Okay, all right. So I put this little thing, that little thing right there, you'll see them in a couple of slides here, means that there's some important point across from it here. So right here, these little spicules here is, you see where it says multiple individual trabeculae enlarged by appositional growth. Appositional growth means they grow from the outside. All right? It's called an intramembranous process, but it's by appositional growth. Although primary bone tissue formation begins as an interstitial process, once it grows up, it becomes appositional. And type 1 collagen is the main factor here for bone. And this is what it looks like. If you take a section through a bone, here are the spicules that are joining, that have joined and are grown. Between them is the bone marrow. Now, one thing you need to keep in mind is that you see here a surface of osteoblast. Up here you have a surface of osteoblasts out there on the surface. Remember that this is going to be called the periosteum. The periosteum is the outermost layer of the bone where reside the osteoblasts. And this is what you got. This is all the cancellous bone. You can see all of these things right here, translate them into a real bone, and you can see the lines of tension or stress, how they're arranged here. This is spongy bone or cancellous bone, and here's the compact bone on the outside on both sides. It goes around the collar of the bone. Now, the compact bone operates by a different mechanism. It's not that interstitial bone growth that we saw earlier. It grows by a process called Haversian systems. So it's 10 till, take a break before we get into how these